Thank you for participating in the NIH Metabolomics Scientific Interest Group webinar series. Today's webinar from Dr. Tom Metz will focus on shifting the metabolomics paradigm, exploiting computationally predicted metabolite reference data for comprehensive metabolomics. Before introducing our speaker, I'll quickly review a few logistics. Should you experience technical difficulties, you can contact us through the questions box in the expanded control panel, or by phone or by email. If you need to view live closed captioning, please click on the link that will appear in the chat box. At the end of Dr. Metz's presentation, we will allow time for questions. If at any time during the presentation you have a question, please type it into the question box and I will ask it on your behalf during the Q&A session. So it is my pleasure to introduce Dr. Tom Metz. Dr. Metz earned bachelor's degree in biology and chemistry before earning a PhD in chemistry from the University of South Carolina, where he studied the role of Meyer chemistry in the development of diabetic complications via the chronic cumulative chemical modification of tissue proteins. In 2003, he joined Pacific Northwest National Laboratory for postdoctoral work in mass spectrometry with Dr. Richard D. Smith, where he focused on metabolomics. He became a staff scientist and a principal investigator in the Integramomics group in 2005 and is the metabolomics lead for a group of scientists that focuses on development and application of high-throughput metabolomics and lipidomics methods to various biological questions. Dr. Metz's research has focused primarily on applying MS-based omics approaches, including proteomics, in studies of diabetes mellitus and infectious diseases. This has resulted in over 150 publications to date. Currently, he is a director of the Pacific Northwest Advanced Compound Identification Corps within the NIH Common Fund Metabolomics program. So we're looking forward to hearing from Dr. Metz today, and welcome.
Thank you, Krista. I appreciate the invitation and the opportunity to speak today. Hopefully everyone can hear me. I'm about to share my slides, so oops. Let me know if you cannot see those. I will move this little panel out of the way a little bit.
Tom, we can see them and we can hear you well, so you're good to go.
Yep. Again, want to thank Krista for the opportunity to speak. I'd also like to thank Bryce, who's our technical support person, who's been very helpful in working the logistics here. As Krista mentioned, I'll be talking about what our vision in our current research is at Pacific Northwest National Laboratory, but also in the PNA-CIC, which is the Compound Identification Development Corps funded by the NIH through the Common Fund Metabolomics program Phase 2. And really what we're focusing on is exploiting predicted reference information, tandem mass spectra, CCS values, and so forth, anything that we can get a hold of and that seems to have good predictive accuracy, and to use that for confident metabolite identification. And I also thank you as the attendees for connecting, and I hope you're all staying safe and healthy and sane in these challenging times. And speaking of which, I just wanted to lighten the mood a little bit and ask you to vote whether you like Gary Busey Tom or Babyface Tom. So the photo on the left was after several months of not having a haircut or shaving after we went into our stay at home situation in Washington State. And then finally my wife said, you have to clean up. And so I bought a pair of clippers and she cut my hair, and now I look, according to all my friends, 10 years younger than what I did before. So cast your votes, and I'd be interested to see what you think. So this is the outline for the talk. I first want to talk about my personal experiences that have influenced how I think about metabolomics. I think that's an important thing to do in general in life, to try and understand where another person's coming from before you pass judgment on them or what they're doing, and especially in today's times, I think it's very much needed. I'll then talk about what we're doing, the work that we're doing under our PNA CIC core, and then I'll show some initial applications of the use of Insilico reference libraries for compound identification. So from some background perspective on things that I've experienced through my research up until now, I performed graduate research studying Maillard chemistry, which is non-enzymatic chemistry, and it was first characterized by Louis Maillard around 1912. He did some experiments heating glucose and glycine, and then looked at the products that were formed and characterized these as brown pigmented products or melanoidins. And then subsequent work through the years further characterized the detailed molecular chemistry that occurred during that heating of glucose and glycine, and resulted in the elucidation of the first stable product of that reaction, which is known as the ambidori compound. And this type of chemistry, Maillard chemistry, also known as Browning chemistry, is very important in food chemistry, because these products lead to the compounds that affect the flavor and the aroma and the texture of food. And so all that research was fascinating, of course, to learn why food tastes the way it does. But really, it didn't get, that exciting for the research community until this type of chemistry was shown to have relevance in vivo. And in 1968, Samuel Rabar, who you can see here, was doing some basic studies of different types of hemoglobins, different isoforms that were present in certain populations in the Mideast, and he identified a fast-moving form of hemoglobin that eventually was characterized as HbA1c and was found to have a molecule of glucose bound to the beta chain amino terminal valine residue of hemoglobin. And today, HbA1c is actually used for long-term monitoring of blood glucose control in individuals with diabetes. It's in most of the kits that you have for long-term assessment of glucose control involve looking at HbA1c. So I found it fascinating that this non-enzymatic chemistry that occurs when you cook food also occurs in people. And my graduate advisor, John Baines, at the time said, well, people are basically ovens. They just have a lower temperature but a much longer cooking cycle. So, these things began to make sense. And in fact, we become browner as we age. And what you see here are costal cartilage samples, the cartilage that are between the ribs. And you can see that as you age, this cartilage becomes darker and darker and browner and begins to take on the characteristic pigments that are very typical of Maillard chemistry. And so essentially the human body is accumulating these products of non-enzymatic reaction in long-lived tissue proteins, those proteins that don't turn over very frequently. And part of my project was to assay for a small molecule inhibitor of these types of reactions and to look for reactions of that inhibitor with molecules that had free carbonyl groups, aldehydes and ketones, and then to quantify those in the urine of diabetic animals. And so that was my first adventure into small molecule characterization, and I really had a strong appreciation for the role of chemistry as it affects biology coming out of that research group. So then I moved to Pacific Northwest National Laboratory. I'm in my 17th year now here, and at P&L I focused on mass spectrometry in general, but in terms of specific applications, it was in untargeted omics measurements, metabolomics, but also including proteomics. And today I lead a team within our larger group, the Integrative Omics Group. It's about 80 people, and my team focuses on metabolomics. And while at P&L, I also had a strong appreciation for the finer points of different types of instrument development, whether it's on ion optics, like you see here with the ion funnel, or LC developments. These LC systems were all high-pressure systems, and they were developed several years before Waters then commercialized the UPLC system, and other companies have come on now with their own high-pressure systems. We've also developed software for treating the data, and now the group largely is moving more and more towards gas phase separations, I-mobility, and the IMS work has predominantly been led by Dick Smith, but also others in the group here as well. Which brings me to metabolomics and the current grand challenge of metabolomics. You might hear Lloyd Sumner give talks, and he'll talk about the grand challenge of metabolomics, and it's really unidentified metabolites. And I really love to use this example because I think it really makes the point of this challenge that we have. This is data from the Undiagnosed Diseases Network, where we were the Metabolomics Core from 2015 to 2018. And the purpose of this UDN program is to perform different types of assays on individuals who have rare disorders that have yet to be characterized by medicine. And so we perform metabolomics analyses, and the gray dots that you see here are reference data from our analyses of about 120 people that had no known metabolic disease that we used as background information. And then we have metabolomics data from a patient in the UDN, indicated here by the proband, and then that patient's mother and father. And so you can see how far beyond the average metabolite relative abundance certain metabolites are for these three individuals. But I draw your attention to a metabolite here that was very, very interesting, that was 4 Z-score beyond the average profile for that molecule in the background data, and it was high in all three individuals, the patient, the mother, and the father. And so in the context of the Undiagnosed Diseases Network, we were very interested in this, but then you scroll over, and it's a metabolite that we can't identify because we don't have reference information for it in our reference libraries. And so very frustrating. You know, we don't know if this is indicative of the person's disease. We don't know if it's because all three individuals had the same thing for breakfast. We don't know if they got new carpet in their house and the carpet's off-gassing and they're accumulating these things off-gas. Very frustrating. But this is pretty typical of a metabolomics study where you usually, depending on technology used, usually identify less than 10% of the data that you can detect. And usually a large majority of things that you can't identify happen to be the things that have high fold changes and low p-values. So it's frustrating to have such obvious information tied into phenotype that we can't identify. But this is really a characteristic of our current metabolomics paradigm and how we actually confidently identify molecules in our high throughput screening studies. And the way that we do that now is we analyze pure substances that we buy from chemical suppliers. We send it through our instrumentation. We measure, the properties that we're interested in, and we put that information in a database. Then we analyze our samples of interest, clinical samples, for example, on the same instrument platform. We take experimental data, we match it to the library, and then things that match well, then we assume it's a confident identification. So the problem with this entire paradigm is that typically any laboratory has a few thousand pure substances in-house that they can use to build their libraries. And so we, as metabolomics scientists, pretty much identify the same molecules over and over again because those are the same molecules that we can buy from chemical suppliers. If you look through the literature at some of these analyses of what we call chemical space, the expanse of chemicals that are thermodynamically stable that might appear in nature, then some estimates put that at anywhere from 10 to the 30 to 10 to the 60 compounds, and a lot of those are based on combinatorial approaches. But the point being, there's a lot of chemistry out in the world that we have an inability right now to confidently identify in our high throughput screening studies. So I want to contrast this to a typical proteomics study. I've conducted a number of proteomics studies in my time here at P&L, and so I love to draw this contrast. So we can take protein samples from any type of tissue or biofluid. We send it through our sample processing and analytical pipelines, and we can generate really a lot of data. And in the experiment that I just showed you previously, which was a study of human islets, stimulated with cytokines to induce a stress response as a means of identifying potential biomarkers. We generated almost 2 million tandem mass spectra. We searched all of that data in 3 to 4 hours, and this will depend on your computational capabilities and the number of servers and things that you have available to you for housing proteomics processing software. We identified 650,000 spectra. It's about 36% of the data, a little bit better than metabolomics, but still not, the majority of data being identified. But importantly, we identified 30,000 peptides that map to 11,000 proteins at less than 1% false discovery rate. And from there, we went on and performed additional follow-up experiments, mouse models, so on and so forth, and we actually identified a novel protective factor of beta cells in vivo. It wasn't a novel protein, it had been known before and characterized before, but the role of that as a protective factor had not yet been characterized. And I can tell you that we looked at 0 raw data from this proteomics experiment. We trusted the computational pipelines and the methods for limiting false discovery and moved on directly into our biological interpretation of the data. And this does not happen right now largely with metabolomics. We generate our data and we spend a lot of time going through and curating the data to make sure that we're not making misidentifications. So this leads me into our core project that Krista mentioned and what our vision is for the future of metabolomics. I truly believe that relatively soon we're going to get there as a community, but I'll tell the story and we can discuss it later. So we're one of 5 compound identification development cores that are funded under the Phase 2 Common Fund Metabolomics Program. You can see us here in Southeast Washington State. The stars indicate the other CIDCs in this program, and the plus signs indicate some data and software development centers. And you might recognize other entities that have been existing in the metabolomics program for quite a while, such as the Metabolomics Workbench at UCSD and Shankar's lab. This is what our structure looks like, and this is actually the structure of all of these cores as required by the call that we responded to. There's a computational core, and there's an experimental core, and there's tight coupling between the two. The computational core predicts molecular properties, the experimental core measures those and feeds that information back to the computational core so that the predictive algorithms can be refined. And as part of our team, we have David Wishart, who's participating and providing access to the CFMID algorithm updates, as well as biotransformer, which predicts metabolism for metabolites of interest. So our motivation was basically, can we answer 2 questions? One, can we find a methodology that allows us to confidently identify a molecule without relying on data from authentic reference materials that you purchase from chemical suppliers? Also, can we identify molecular properties that facilitate these approaches for confident identification and in a high throughput, that is, automated pipeline? And the 2 questions are related, and so the 2 answers will be related. So for me personally, the inspiration came from proteomics. You may or may not be familiar with it, and so if you're not, I'll provide a little bit of a background using this cartoon. We take proteins, we proteolytically digest these with an enzyme. Usually we use trypsin because it puts them in pieces that are very amenable for a reverse phase liquid chromatography separation, and also nice size for tandem mass spectrometry. So we perform separations just like what we do with metabolomics. We perform MS-MS just like what we do with metabolomics. But peptides fragment very, very characteristically, so that it allows us to predict what they should look like, and then makes identification, comparatively easy in comparison to metabolomics. Why is that? Well, proteins and peptides have certain characteristics. Proteins are direct readouts of the genetic code. So if you know the genome of the organism from which your sample came from, then you can predict which proteins you should get. We're not necessarily there yet in terms of being able to do that type of prediction of the metabolome in an organism confidently, but there are a lot of good work, lots of good work through the community that is leading to that, but I think still a little bit more to be done. Peptides also fragment very characteristically around the amide bond during MS-MS. So you see the mock peptide down here, and you can see that you might get A or X ions depending on the fragmentation mechanism that you're using. You might get B or Y ions. This occurs during CID and HCD type fragmentation. You may get C or Z ions. This occurs during electron transfer dissociation and so forth. But the important thing is that the fragmentation always occurs in this region of the amide bond. And so that has allowed the development of algorithms that can computationally predict what a peptide dissociation should look like. That enables, essentially, confident identifications of peptides without the use of authentic reference peptides. And there are also good methods for assessing false discovery, and I'll show that in the next slide. But essentially, when one performs an untargeted proteomics experiment, there's really never any type of follow-up using standard reference peptides to confirm the initial identification in the discovery experiment. We may indeed do validation studies later in other cohorts using a targeted proteomics experiment to verify the findings in a replication of the entire experiment. But there's never an identification or a comprehensive validation of the initial discovery data set. We rely on the paradigm and the predictive software that have been developed for processing the data. There are also very good methods for assessing false discovery rate with proteomics. This isn't the original paper on the target decoy database approach, which was developed by Steve Gee, but this is a nice cartoon to show you that if you give the search algorithms decoy data, and we create decoy data by sometimes reversing the protein sequences or scrambling the protein sequences that we're searching against to create essentially fake data. And then we look at the proportion of the data that matches to the target database versus the decoy database, and you very often see these nice distributions, and you can draw essentially a line down through the score on the x-axis and cut off all of these false hits by this sliding scale of where you want your false discovery rate to be. So there have been a few papers out there on exploring false discovery rate methods for metabolomics, but we don't have anything yet that has been widely adopted by the community, so a little bit more work to be done there. So coming back to this slide on the motivation, we added 2 requirements to what we're looking for in an answer, that methodologies will involve some type of a priori assumption of the molecular characteristics or behavior of the molecules that will allow confident and accurate predictions of these, and that these properties should be definitive, meaning unambiguous when performing identification, and experimentally measured and accurately, experimentally measured in an accurate and reproducible way. So our aims for this Pacific Northwest Advanced Compound Identification Core is, 1, to generate methods that lead to confident prediction of metabolite properties that we can then use to generate Insilico derived reference libraries, and then 2, to develop and apply a multi-property matching approach to utilize all of these different properties that we are predicting in a confident way, and then 3, make all this available to the research community. And so what we envision for the new paradigm is the cartoon I showed earlier. We replaced the upper half, which is library building, and instead of using or performing analyses of pure compounds that we've obtained from chemical suppliers, we predict the properties that we would like to measure. But this assumes that we can do so accurately. So I just wanted to point out we're not the only group doing this. There are many others that are developing predictive capabilities, whether it's on tandem mass spectra prediction or retention time prediction or prediction of collision cross-section. So, it's not something that we thought of de novo. There are many others that realize this challenge in metabolomics and that are trying to address it. And I think ultimately we as a community will be able to take the best of all of these types of predictive algorithms and combine them in a comprehensive approach to generate libraries. I'll just mention that our approach is based on ion mobility spectrometry. Ion mobility has a lot of nice features, I think. It provides an additional dimension of separation, so that increases the coverage of the metabolome by sort of inline fractionation, increases the dynamic range of the measurement. It's hooked up to a mass spectrometer, so of course you get mass and isotopic distribution information. You can also generate tandem mass spectra as you normally would. But importantly, you can measure a physical-chemical property of a molecule called collision cross-section, and I'll talk a little bit more about that in the next slide. And I will mention also that ion mobility has a nice ability to separate isomers and isobaric compounds that liquid chromatography and a mass spectrometer typically cannot. And so that's part of why you have increased dynamic range and coverage of the metabolome during measurements. And I personally like to measure as many properties of a molecule in a single experiment as possible, and you've probably seen this typical cartoon where blind scientists are touching different parts of an elephant and they think it's different things. Well, if you use only a few properties, you might have a similar experience with a metabolite, and you might think it's a certain metabolite because you're seeing a certain point of view of the molecule, but you're not getting the entire picture, and so you might make a misidentification. And I feel the more views that we can get of a molecule, then the more confident and sure that we'll be in the chemical structure. Just a little bit about collision cross-section. I'll point out three things here. The collision cross-section is not influenced by chemistry, so to speak. And what I mean there is that when you're performing liquid chromatography analyses or gas chromatography analyses, you've got partitioning of the metabolite or molecule between a stationary phase and a liquid phase. With ion mobility, the molecule is already in the gas phase in the instrument. Really, your separation comes down to the structure of the molecule, the ionized molecule itself, and the voltages and temperatures and pressures that are present inside the instrument. So you've got more physics impacting the readout in an ion mobility experiment and less chemistry. And so you don't have degradation of stationary phases and so forth that cause shifting and retention times. So what that means analytically is that you can obtain highly reproducible measurements in your own laboratory, across different instruments in your own laboratory, but also across instruments in different laboratories. And there was a nice paper that I mentioned down here in 2017 where 4 laboratories participated in the measurement of the same compounds, and the relative standard deviation using the same instrument and the same operating protocols to collect the data, they obtained an RSD of less than 3.3%. It was fantastic. CCS is also a property that can be accurately calculated using quantum chemistry from first principles, and it can also be predicted using machine learning approaches, which I'll talk a little bit coming up. In our core, we use 2 different tools to predict collision cross-section. One is based on quantum chemistry, that's shown over here on the left, Insilico. The other is a deep learning-based tool, and both of these tools were developed by Ryan Renslow here at P&L and his team. And you'll see the errors that are generated through these tools when comparing predicted CCS to experimentally measured values. The measured values are around almost 2,000 different conformers. And you also see the computational time required for some of these. So if you can get by with less error, then you can generate predictions relatively quickly. If you want really, really accurate predictions, then you're going to pay for it in terms of computational time. And these are node hours on the supercomputer here at P&L. We're also collaborating with Dave Wishart at the University of Alberta on this project, and Dave is lending his expertise in MS spectral prediction through the CFMID algorithm. Both of these CCS prediction tools have been published. I'll just step through this cartoon to show you at least for the quantum chemistry pipeline, how it works. You give it molecular information in the form of an INCHI. You can also use SMILES. It will convert that to a three-dimensional chemical structure that's readable by a computer. We use molecular dynamics to model various gas phase conformers. Those are then processed through density functional theory to optimize their structures. We generate collision and cross sections for these, and then we use Boltzmann weighting to arrive at one probable collision cross section. Move on here. And just an example to show that this has worked very well in a demonstration case, looking at cis-trans isomers of a molecule that we call diCQA, dicathylquinic acid. This was a study conducted by Ryan Renslow and Aaron Baker at PNNL. diCQA is an antiviral compound that usually occurs in one particular form once it's isolated from the plant, but then if it's exposed to UV light, then it begins to isomerize, and these are difficult to separate using conventional approaches, but using ion mobility, and in this case, this is data from structures for lossless ion manipulations that I didn't provide much background on, but it's a very high gas phase resolution technique. And you can see the predicted values of these cis-trans isomers agree very well with the experimental values. And in fact, we observe around 0.8% average error compared to the experimental data. But again, this was with the most accurate version of our quantum chemistry pipeline that takes a long, long time. So we tend to accept higher error and use the higher throughput techniques to generate our reference information. And so far, for CCS, we've generated values for about 100 million compounds, and these are all available at this website. You can check it out and try it and see how well it works. And there are data there from the different flavors of the two different predictive platforms that we have. The majority of values that are up on the website right now are from our dark chem deep learning approach. And then this just showed some updates from CFMID that you'll be reading about soon. I think David's lab has a paper in review now there on CFMID, next generation CFMID. So I'll probably move a little bit more quickly here through these initial applications of the use of Insilico libraries. The first was our participation in the EPA ENTACT program, where the EPA provided different laboratories and chemical mixtures in a blinded format, and the laboratories that participated were allowed to use whatever technique they wanted to try and identify the composition of the mixtures. We used a combination of ion mobility and Fourier transform ion cyclotron resonance mass spectrometry, and generated predictive data. We didn't use data from analysis of authentic compounds. We used a reference library that was entirely comprised of predicted values. So we predicted CCS and isotopic signature and mass, which are more or less trivial predictions compared to CCS. And our results were, good and bad. The good part was we had an accuracy of 96% in determining the correct composition of the mixtures, at least for things that we were able to detect. The bad news was that the false negative rate was high, the false discovery rate was high. But importantly, it showed promise and gave us encouragement to continue down this path. And also a good sign was that as we increased the score that we came up with, a total confidence score, then the false discovery rate dropped, which is what you would like to see. And that paper has been published as well. You can read the details there in the Journal of Chemical Information and Modeling. I'm going to skip through these slides a little bit in the interest of time, just so that we can get through to the Q&A session. But I wanted to show some data of our initial applications of the ion mobility platform to a study of sleep deprivation or altered circadian rhythm in study participants. And this is a collaboration with Hans von Dongen and Shoban Gadamiti at WSU. And it's just briefly on the study design, we've got people that are on simulated night shift work, people that are on day shift work. The people that are on the night shift are supposed to sleep during the day and be up all night, and vice versa with the day shift. And so we look at the blood to see, what might be going on there. And Hans had already published some metabolomics data looking at these samples with a collaborator in Australia, and they used the Biocrates kit. You can see how many metabolites they detected, how many of those were rhythmic. You can see the patterns here. So definitely there's circadian rhythm with small molecule abundances in individuals, and that can be disrupted when they go on an altered sleep schedule. We did a complementary lipidomics analysis. Project was led by Jennifer Kyle here, looking at the same exact samples, and sure enough, we can replicate the cycling behavior of lipids with time, which gives us confidence that we're able to tease out these differences using a conventional lipidomics analysis. We then applied liquid chromatography, ion mobility, tandem mass spectrometry of the same samples, focusing on the polar metabolome. A lot of analyses. We analyzed each sample in positive and negative ionization. We also used 3 different collision energies for the tandem mass spectra. Resulted in over 916 raw data files, if you include blanks and QCs. A lot of data, about 1.3 terabytes in total. And then we're stumped, really, with how to effectively process that. And so we decided to develop our own software, Deimos, for multi-dimensional data extraction to extract data from the LC dimension, the ion mobility dimension, and the mass spec dimension. So I'm running short on time. I'm going to skip through these slides on Deimos. It's just showing some progress on where we are with developing the software. It's almost finished, but as I step through these slides, I'm not going to read them. I just want to point out that we are able now to extract data, the multidimensional data from that platform. We're able to align it. And so these top-bottom plots here are data from 2 different QC samples that we had interspersed throughout the run, and you can see there's good agreement in the mass spec domain, as you might expect. There's good agreement and alignment in the drift time domain, and a little bit less agreement in the LC domain, but still not bad. And we've fixed some of these misalignments using a SVR algorithm so far. De-isotoping of the mass spectra is an effort ongoing. We're working to deconvolute the data-independent tandem mass specs that are being generated. The instrument itself can do this to a degree, but there's still a need for an algorithmic improvement to the deconvolution. This type of deconvolution is very analogous to what happens with GC-MS data. And you can see the difference for this spectra between a windowed approach, which is what the instrument does inherently, versus what we can do algorithmically. And you can see that the algorithmic approach is removing some ions that likely do not belong to the molecule of interest using just the windowed approach. So that work is in progress as well. Coming back to that sleep study, we've generated a lot of data. Close to 1 million features, 3-dimensional features in positive ionization, over 2 million features in negative ionization, and I'll be honest here and say that we've set our signal to noise fairly low to not miss anything. And we also haven't implemented de-isotoping yet, and so we've got multiple features included here from the multiple isotopes. But importantly, many of these are statistically significant based on the statistical methods that we're using, and you can begin to see some cycling behavior here. The bold blue line and the bold dotted line are the averages from the study participants. So it's nice to see that we're replicating the data that we've seen from the previous publication, as well as the complementary lipidomics analyses that we've performed. I'll just quickly touch on our attempts right now to use all of our predictive data to confidently identify metabolites. So we believe we've identified glycine betaine in these samples. This is just looking at 3 representative data files out of the over 900. We had 15 MSMS spectra matched to experimental spectra in a variety of repositories, mainly MONA and NIST. There were no experimentally determined CCS that we could find for this molecule, and so we matched to our Insilico generated Insilico spectra, or Insilico CCS instead here. This just shows you 2 features that had evidence to support that they could be glycine betaine. They have the same retention time, as you can see here, about 8.25 or 3 minutes, but they have different drift times. So likely what we have here are 2 isomers or isobaric species that happen to have the expected mass of glycine betaine. And so then we move on to tandem mass spectra to try and identify these. And looking at the different spectra that were available through MONA and NIST, you see different degrees of match depending on the collision energy that were used. We had our best matches to these at the higher energies. You can see the dot product scores here. 20 volts seem to do well, and then 40 volts not so much for this particular reference spectra, but for the other 2 reference spectra, one from MONA, one from NIST, we see relatively high scores as well there. And then for the feature 314, actually the predicted spectra from CFMID at 10 volts matched very well, whereas 20 volts and 40 volts not so much. Digging into the details a little bit, it was interesting to see the data from these 2 features, again shown here. This shows you the number of tandem mass spectra that had hits for this feature 314 and their dot product scores. You see the same dot product scores down here for the 331 feature. These are summaries of the previous slide that I showed you. You can see that we had a systemic mass shift in our mass accuracy through the course of the analyses. That's addressable through recalibration of the data. But interestingly, if you go on and look at the CCS error, and again, these are all predicted CCS through our pipeline, the feature with ID 314 had a relatively reasonable match to CCS at about 3.2% error, whereas the feature at 331 had a poor CCS. And so although 331 had a few tandem mass spectra that matched well to experimental data, the predicted MSMS spectra did not match well, and the CCS predictions did not match well. So it doesn't mean that this isn't glycine betaine. I think it means it's not the particular gas phase conformer that we think it is. And so we're realizing that we've got a lot of data to go through, but I think a rich resource to begin to understand the subtleties in gas phase conformations of molecules and how those impact the tandem mass spectra and the CCS and the other properties that can be measured by mass spectrometers. So that was essentially the last slide. In summary, I personally believe that Insilico reference libraries are the future of metabolomics. The proteomics community has been able to do this in part just because of the inherent chemistry of a peptide and a protein. I don't think it's impossible for metabolomics. I just don't think we've arrived yet on the properties and paradigm for how to do this yet, but many groups are working on it, including ours. I think we're going to get there sooner than later, and I tend to be a relatively impatient person, so I'd like to have this capability and use it while I'm still actively doing research. Multidimensional analyses, such as LC, IMS, and mass spectrometry, they provide higher coverage of the metabolome just because of the additional separation that occurs during the measurement. Hopefully you can begin to appreciate that additional analytical properties of the same molecule being measured, for example, a mass, an isotopic distribution, a tandem mass spec, a CCS, they start to provide higher confidence for one identification over another, but it also introduces complications and other considerations that we now have to deal with, and primarily it's how do we integrate and weight appropriately all of these multiple lines of evidence to provide a molecular identity. Something we're working on actively now in our project and our research, and it would be interesting to hear what you might have, what thoughts you might have on this as well. So with that, I'll just thank the project team, our funding sources, the NIEHS and the Common Fund, as well as some internal funding for the data that I showed, and I'll stop now and take any questions that you might have. I see some questions and comments in the chat box. I can start through there.
Oh, geez,
oh,
sorry. I'm so sorry. I was muted, and I apologize, yeah. Tom. Thank you for chiming in, because I would have been talking away. My apologies.
Yeah.
There are a number of questions. First one I'm going to address very quickly. A number of people would like to get your slides after the webinar.
Sure.
We do post those. Tom has agreed to post them, and it takes about 2 to 3 weeks to get them posted because there are some procedural issues, and we will have those posted on the Metabolomics Scientific Interest Group website. And moving on to the scientific questions, the cell metabolism paper looks interesting. I'm wondering how one can select a specific, such as you did, for GDF15 from a large number, over 300, that were found to be different. At the end of the day, experiments have to be conducted to validate the target. So how can confidence in picking, to validate the target, so how can confidence in picking the correct molecules is high?
Yeah, so if I interpret the question correctly, I interpret it as you've identified 11,000 proteins. How do you then down select to where you think the action is in terms of biology? And there are a number of ways you can do that. You can look at volcano plots of the molecules of interest or the data. You can look at the correlation of fold change and p-values and so forth. But for that particular study and that paper, I really have to credit Ernesto Nakayasu here in our group, who is the first author of that paper, and he, I feel, had amazing insights. In his analysis of the data, he looked at additional cytokines that were turned on or off through the in vitro cytokine stimulation of the human islets. And of course, we saw many cytokines that were upregulated, which you might expect as part of the propagation of the stress response. But Ernesto keyed in on 3 cytokines and or growth factors that were decreased in expression. And he had a hypothesis that because those were going down, those might be protective in nature. And we worked with our collaborators, Raghu Miramira at University of Indiana, and we did a lot of follow-up studies on using mouse models. We were looking at pancreatic slices from NPOD, a repository of tissues from individuals with diabetes, a lot of follow-up studies. So all of the follow-up work is really described in nice detail in that cell metabolism paper. But sometimes, in addition to all the normal logical steps that you go through to parse the data to focus in on things that you think are interesting, there's often, what we say is gut feeling and intuition. And I think in that case, it was more intuition, and we sort of got lucky in that it panned out into a nice biological story.
Great, thank you. Next question. It's well known that bilirubin can be shifted through different double bond isomers by blue light. Is this something in metabolomics we should be aware of? Does it occur after receiving material for analysis?
Yeah, it's a good question. I'm sure there are all sorts of artifacts that get introduced into our samples, not just from exposure to light, but, for example, freeze-thaw cycles and so forth. And there have been some nice papers on freeze-thaw cycles in particular that look at metabolite stability. I would argue that we really can't fully address these phenomena or the impact of these phenomena until we can identify the majority of our data. Because when you're looking at, I don't know, less than 10% of the data set as confident identifications and the rest are unknown features, it's difficult to say the degree of impact that any type of manipulation or perturbation of the sample might have on the chemical composition. The proteomics community has had it easy, I would say, through the years, just because of the nature of the molecules involved and the nice tools that have been developed. They can do things like looking at extended gradients and offline fractionation or enrichment techniques, so on and so forth, to see how those types of analytical improvements impact the quality of the proteomics data. And I would argue we can't really do that right now with metabolomics because we just simply can't identify the majority of the data. And so running an extended gradient, doing offline fractionation, looking at effects of freeze thaw and so forth, I would argue that we can't fully address the impacts of those right now, simply because we can't, we just don't have a good, struggling with the right words, a good, I would say, representation of the data in terms of, the total chemical complexity based on confident identifications. We're just not there yet.
Great, thanks, Tom. Next question, how are you planning to validate the Insilico CCSDB with experimental CCS data? What rationale to select compounds and use those available from efforts of other groups' work with IMS?
Mm-hmm. So for the papers that I showed in the CCS section, one was on the Insilico, the quantum chemistry tool, which we call Icicle. The other was on the deep learning tool called DARChem. We had an experimental data set available to us that was generated by Erin Baker during her time here at P&L, and it consisted of, I don't remember how many unique molecules, but it was around 2,000 different conformers of molecules and adducts of molecules, so that would include protonated, sodiated, ammoniated forms of metabolites, all of which tend to have different drift times and a nine-mobility separation, and then correspondingly different collisional cross-sections. So the plan is essentially to generate additional CCS values, experimental CCS values from analyses of authentic compounds. We've got about 10,000 in-house. I'm not sure whether we need the data from all 10,000, but we certainly want to experimentally measure compounds that have diverse chemistries to make sure that we don't have bias one way or the other in either of the predictive platforms, and that's our plan. We've got some molecules that Peter Dorstein shared with us, which are microbial secondary metabolites. We've got molecules from the EPA, which are, these exposure compounds, man-made. They have very different chemistries, halogens incorporated and so forth. And so hopefully, if we analyze a sufficient representation of chemistry and we see good agreement between the predictive tools, then, we'll be much more confident in using them in our routine metabolomics analyses. The rationale to select the compounds is, what I mentioned already a little bit. We don't just want to measure all the metabolites that are present in central metabolism. We just don't want to focus on amino acids, small organic acids, fatty acids, lipids, so on and so forth. We want to get, the gnarly-looking compounds that come from, say, microbial or plant secondary metabolite production, or that come from, anthropogenic sources, flame retardants, pesticides, pharmaceuticals. We'd like to be as diverse in the chemistry as possible.
Thank you. So we have a number of questions that came in. So this is obviously, people are very interested in this area, and I don't know that we can address them all or we might be here till about like 12:30 my time, 9:30 Tom's time. But I'm gonna, I think maybe we can go just a couple minutes over, and I'd like to try to ask maybe like 3 more questions.
Sure.
One came just a little more broadly as a question that I think I saw a number of epidemiologists on the call, so I'm going to ask this one. For the large-scale studies, epidemiologists strongly prefer working only with identified metabolites as it greatly simplifies the overall workflow. Approximately how many years do you think it will be before these methods are available for large-scale studies?
Oh, that's a good question. I mean, I wouldn't limit it to just large-scale studies. I would reframe the question as, you know, when do you think these methods are going to be available, period? I don't honestly know the answer to that. We're working hard on it, as are others in the community. Again, I'll point out that I tend to be an inherently impatient person. I would like to, really grind this out sooner than later so that I can use it. Ultimately, my first degree was in biology. It's because I wanted to do disease research. And then I got into chemistry to better understand the molecular mechanisms of what occurs in the context of biology. And so, although I'm conducting analytical research, although I'm in a large analytical mass spectrometry group, I at the end of the day, mainly care about, what these molecules mean, what these changes in the molecules mean in the context of the biology. So I definitely hear, the intent of the question, and I definitely want to get there and have a method that works well in terms of not only comprehensive identification, but also assessment of identification false discovery rate. I want that like yesterday, but it's hard. We're sort of trailblazing here. I would like to see some reasonably good method within the next 5 years. Again, mainly so selfishly, so that I can use it myself in my own studies and not be frustrated by where we are with metabolomics relative to where we are with proteomics and transcriptomics, for example. Well,
a follow-up to that question, in a sense, even though it was 2 different questions, but I think it might play nicely into kind of what you've just said, is with many different research groups taking the lead on developing in silico reference libraries for each measured property, how do you envision the community coalescing on the best final tools and then disseminating them to the broader metabolomics community? It looks to me like it will be a leap to get everyday metabolomics scientists to use these and not just the developers themselves.
Yeah, it's another good question. I look back at what occurred in the field of proteomics. It was, I think, the early 90s when John Yates and Jimmy Eng developed the sequest algorithm to identify peptides with confidence and relatively high throughput based on the match of predicted tandem mass spectra to observed tandem mass spectra. And I mean, it seemed like like a light switch in terms of the community adopting that at that time, because there was nothing else really that worked as well. And it just progressed from there. There are many algorithms now on the proteomics side and many software tools to identify peptide sequences based on their tandem mass spectra. They all kind of work about the same. There are certainly differences between the algorithms that would give you slight differences in the peptides identified, but at the end of the day, you kind of get the same data regardless of the algorithm that you use, and you get about the same confidence because of the target decoy approach, again, that was developed by Steve Giggy. So I think really what it's going to take for the community to adopt all of these, to coalesce on a method is, you just have to demonstrate it. It'd have to be some well-characterized sample that's maybe handed out to all the different laboratories, and we all participate in a ring trial or, other type of interlaboratory comparison, and, we provide the results, and, hopefully one or more different methods will, generate many confident metabolite identifications with associated low false discovery, and I expect everyone to kind of switch over to using that. I mean, I'll be honest and say if tomorrow such a method came out, I'm going to use it. if we evaluate it and it works well in our hands and we trust it, and it's not a black box of algorithms, we can see the details algorithmically of how the sausage is made, I would use it tomorrow. Again, my focus really is, what does the data mean in the context of biology? It doesn't necessarily matter to me how we get there, as long as we get there in an analytically accurate and robust manner. So I think once a method or paradigm is demonstrated to work and work well, I just see the community naturally adopting it.
Okay, we're going to do one last question here. CCS seems like a great measurement for in silico predictions because it is reproducible across different instruments and different labs. Retention time and MS fragmentation, which are still the most common measurements because IMS instruments are less common, unfortunately, and very variable. What is your perspective on the future of in silico predictions for reference libraries on these measurements?
Well, I think they'll still be used. I've had similar conversations with Oliver Fien and others that are also PIs under the NIH Common Fund Phase 2 program. I don't, personally, I would put more weight on evidence that come from multiple orthogonal properties than I would in one or two properties. And so then I would assume in that multi-dimensional, multi-chemical property space that we're using to characterize a molecule, that you could get by with larger errors in one individual property using that aggregate of different chemical properties than what you could get by with if you were only using one property. So, for example, if I had a CCS error of 5%, and I was only using CCS, then I'm not happy with that 5% error. I would want it below 1%. But if I have CCS and say my error is 2 to 3%, which most of the predictive methods seem to settle in on about 2 to 3% error versus experimental data. If I've got MSMS with a certain accuracy, if I've got retention time prediction with a certain accuracy, if I've got whatever property is being measured, if the errors are a little bit higher in each one of those individually than what I would like, what I would hope would be that the aggregate of all of those properties would be, relatively strong in terms of being able to say that molecule is that, or that feature in the data is this molecule, and to do so with confidence. I believe that will be the case, but we need to do additional work. This is what we refer to as chemical space. You've got this multi-dimensional space of these many properties that you're measuring, and you've got, say, a point out there in that space. I personally would like to know, well, I think, this particular point in this multi-dimensional space is this molecule. I want to know what are the next closest points around that point in that multi-dimensional space. Maybe it's a big gap, even with the relatively high errors in retention time or MSMS prediction.
All right. Well, thank you all for your attention today and your active participation in our webinar. We would like to take this opportunity to thank our presenter, Dr. Tom Metz, for an excellent presentation. And this may have been one of the most enthusiastic level of questions I've gotten in a long time. So there's obviously a lot of interest in this area. So that was a lot of fun to see. So having said that, we would welcome your feedback to inform future webinars. And again, thank you so much, Tom, and thank you all for attending today. Everybody have a great week.
Yeah, thanks for hosting and thanks for attending. Thank you.